Wait.
Our presentation is Discoids Dance with Wavelengths, and our research team is Scott, M, Makia, myself, Maxwell, and Brandon. And this is a potential title for the paper that we do at the end of the project for a catchy title, Panic at the Discoid, How Different Wavelengths Affect Discoid Activity Levels. Our objective is to record and analyze the differences in behaviors of discoid cockroaches across varying wavelengths of color. Our key terms of the arena, we're going to be performing the experiments, infrared light and ultraviolet light. Our prior knowledge is based off of a Turkestan study that had findings of the species being repelled by blue light and attracted to red light. Our experiment will be conducted similarly, but we'll use even more colors and wavelengths of light.
All right, here we have a graph of the visible light spectrum. Notably, the higher the energy, the lower the wavelength, and the darker the color. Also important to note that the visible light spectrum that we can see is only a small sliver of the full light spectrum. There is also the ultraviolet light, which we cannot see, but pollinators and cockroaches can see. Continuing on with the literature review, I want to share a few interesting studies that highlight certain animals being attracted to specific wavelengths. One study conducted in China set up a large catch net and an LED spotlight to determine if nocturnally migrating birds were attracted to any specific color. They found that blue light dramatically increased the number of birds that they caught. A similar study found that pollinators tended to seek out flower patches of specific colors. As you can see from the graph, bees were attracted to purple, whereas ants and wasps were attracted to yellow and ultraviolet lights and flowers. Notably, young, inexperienced pollinators showed the same preferences, indicating that this might be an innate trait. We also found that current pest control techniques are already exploring this observation that flies tend to be attracted to blue light. For example, that is a Dynatrap, I believe, and it is used to attract and kill flies. A team of researchers studying biting flies, in an attempt to explain this occurrence, wanted to figure out why it happens, and they concluded that lower blue wavelength light highly resembles animal hosts and therefore potential food resources. All of this supports the claim that certain animals can be attracted to certain wavelengths. Now, how do cockroaches perceive this light in their environment in the first place? Well, it is all part of their biology and their body plan. Their body plan is very foreign to ours. Uh, they it may be more complex than you may think. It's definitely more complex than I initially thought. The central nervous system consists of the brain, a large bundle of ganglia in the head, and two ventral nerve cords that run down the length of the body. The peripheral nervous system consists of nerve bundles branching from the two nerve systems, or the two nerve cords to the rest of the body, three in the thorax and six in the abdomen. These give the cockroach a form of distributed cognition, allowing them to use information from their legs and other organs to inform the decisions that they make. Zooming into the head, this diagram shows the antenna, which provide tactile, thermal, and olfactory sensors. Vision, which is the focus of our study, arises from the compound eyes, shown here in the black ovals. They have roughly 2,000 ommatidia, which are the photoreceptors that receive light to create this mosaic vision. A diagram of that can be seen on the right. These images are panoramic but relatively low resolution, so they do provide color vision as well as the UV spectrum. The two ocelli, slightly lower in the white ovals, are not used to create pictures, but rather process slight changes in light intensity, which aid body orientation and navigation. This is especially important as they are nocturnal creatures. Althaus and his team created a 3D map of the cockroach brain you can see on the right there, and you can see how complex it is. Information from the compound eyes travel along the optic nerve to the optic lobe, which processes the visual signals. So that's moving down these yellow, white, and red structures. Then the signals move to the mushroom bodies, this clump of green, blue, and purple structures. They play a role in multisensory processing, learning, and memory formation. Overall, this shows they can't be purely reflex and fixed action pattern based creatures, but can make decisions based on their environment. We are interested in determining if this affinity for darker colors, like the flies, is also true of discoid cockroaches.
So for our research question for this project, we have how do discoid cockroaches respond to varying light stimuli across the different wavelengths of color? This includes from the blues and ultraviolets to the reds and uh, infrareds. Um, our alternative hypothesis is as wavelength of light decreases, the cockroach activity level will increase because they're accustomed to darker environments where there are less risks of predators and more food sources available. So um, we believe that the different wavelengths of light might change their activity level just based off of that there's not much light in these darker environments. And then for our null hypothesis, we have as the wavelength of light decreases, the cockroach activity will not show any change.
So going into our materials and methods, first we'll start with the materials. Pretty simple. We'll be beginning with a big bucket, and that's what we're calling like our arena for now. It's important that we have a lamp with red, green, blue light, UV bulb. We'll have a ruler to record walking distance. We're going to use gloves for interacting safely with the cockroaches. And then we'll have a stopwatch to record time. We want to make sure that we're consistent each time during the recording of behavior. And then a big thing is going to be the cockroach ethogram. And I can talk a little bit more about that. It's basically going to be measuring escape behavior. And so we're trying to make a scale between 0 and 5 so that like every minute we can mark what behavior we've observed and like have a numerical value to put that with. And so 0 would be cockroach doesn't show any response to the stimulus. So it remains still, where 5 would be all the way completely demonstrating the escape behavior by hiding in like the things that we'll be providing for them. And so if you go to the next slide, it talks a little bit about what that would look like. So we'll start by putting the bucket in the middle of the room. We'll line it with an even spread. We're saying 2 cups of substrate, so we'll make sure to do 2 cups every time. And then we're going to have a toilet paper roll on each side of the bucket to provide potential hiding spots. And this is really important for the ethogram because if it's a 5, then they're going to be using those toilet paper rolls we provided. And if it's a 0, they don't interact with them at all. And then we'll position the light source on the chosen side of the bucket, the arena. Both left and right sides will be tested across trials, but we're going to alternate. So we'll do each individual on the left, and then we'll rotate and do each individual on the right so that they, instead of doing one individual left and right, one individual left in, right? So that way it like gives them a chance to acclimate some more. For the control testing, before introducing any light stimulus, we'll record the cockroach behavior in the bucket with no light for comparison. And the way the procedure will be we put in one individual, we allow them to acclimate for 10 minutes, because it would be hard to say that any behavior immediately after putting them in there is a direct correlation to whatever stimulus we're introducing or taking away. So they need a moment to acclimate. And then using gloved hands, we'll place the test subject in the middle of the bucket. We'll turn on the light source and measure the cockroach's behavioral responses according to that predefined ethogram. And we'll record behavior for about 5 minutes. It's important not to do it for too long, but also not too short so that we can repeat it multiple times. But also that way we know we're not just seeing something once or twice and trying to draw a conclusion from that. And then we'll repeat the procedure with each individual, rotating the location of the light, as I said earlier, to eliminate any bias. And then different light sources will also be tested, specifically the wavelength frequency. And we're going to be testing all different visible light spectrums. And then the variables for this experiment, our independent variable is the form of light and the frequency of wavelength. So if we do UV light versus visible light, and then our activity level index is the dependent variable. So the ethogram, what we can record, what we can measure. And the controlled variables are things that we need to keep consistent. So amount of bedding, light intensity, interval time, number of the toilet paper rolls, the starting location, they have to be in the exact same spot every time. And then temperature and humidity as well. We can't always make those exactly the same every time, but it's important that we take note of it when we do, and that we want it relatively around the same. And then as well as the time of day, we need to be consistent with that because these are nocturnal animals. So if we get closer towards evening, it's likely they may demonstrate behaviors that maybe they wouldn't have if we did the experiment earlier the previous day. And the control group will be the absence of light. So no light to see a baseline for behavior.
For our statistical analytic plan, for our hypothesis testing, we are going to set the cockroach 40 centimeters from the light, measure the distance traveled, compare the distances, and review and compare the ethogram results. We're also going to calculate the standard deviation to understand how dispersed data sets are in relation to the mean. For our broader implications, this fits into the broader literature of animal studies because of the sensory inputs on behavior. As we know, these cockroaches aren't studied a lot. And so previous research has explored light sensitivity with these and circadian rhythms in various species, but it's filling a gap because their visual processes could have broader implications for understanding how insects and invertebrates adapt to light in their natural habitat. Again, as mentioned before, these are nocturnal animals, so seeing how they respond to that is important. Continuing, how is this study unique and important? It studies how different wavelengths of light and color impact the behavior of cockroaches and the effect of light on nocturnal, less visually reliant species like cockroaches have been understudied. This also could have a practical application, such as pest control and ecological studies. This could be maybe a type of repellent used that's more natural and non-invasive as chemicals. Lastly, here are references. Thank you.